Hey guys, it's Martin Shkreli with part 2 of the Axon Clinical Trials Analysis series. So I think where we left off is we're looking at press releases and SEC filings, so we might as well continue to do that. And what we'll also do is get to a PubMed search of the drug as well. So let's quickly kind of type in the stock ticker and SEC filings, and I think it makes sense to read every single one of these. It doesn't look like there are too many. I don't know, 40 here. Let's put it up to 100. You go back 100, you pretty much are back to the start of the company. So you can see the S1 and so forth. So you're basically brand new at that point. So from 1 to 100 here, draft registration statement, confidential registration statement, and so forth. And eventually the company goes public around 2015. Let's see, it's the S1 when effective, right? 610. So jot that here somewhere. 610, 2015. Company goes public. And actually, while we're here, let me just go jump on and make sure that the folks who want to see this live can see it live. Reminds me of Bill O'Reilly. We'll do it live. We'll do it live. This will just take a second. All right. So hopefully that'll work out. Registration of securities. So what is this Form 8A? Not very common. Pursuant to Section 12 or Section 12B or 12G. So let's see. Trying to register. Oh, I see. This is a form for moving from NASDAQ or NYSE to NASDAQ. That's pretty interesting. So you'll never see that form again. We knew that form would be the case. Form 25 is the same type of form, right? We are no longer on New York Stock Exchange, so that's Form 25, and Form 8A is to move the securities to the NASDAQ. So those are pretty basic, right? So let's look at this 8K. There's the equity incentive plan. We should probably read that, actually. But let's see. So there's 20 million options now. That's quite a bit. All right. I don't spend too much time on stuff like that, the compensation plan. So they're moving to NASDAQ. We need that. Got a lot of filings related to that. And I wanted to just get the 10Q down real quick. So it's funny because sometimes they say they're in Basel, sometimes they say they're in Bermuda, and sometimes they say they're in London. So it's a little tough to figure out. Let's write that down. London, Bermuda. Which is it? New York. All right, so we have the balance sheet real quick. Cash, I think that's it. And I just like to write over there what quarter we're in. And on the front page of the filing, you'll see the shares outstanding. Oops. 107, 525, 925, you multiply these two. I think they have a little bit of debt. 51. 752 and 4466 due to Roivant, which is sort of the parent company, not exactly the parent company. And there's some beginning of a model here, so we can do that as well. So there's no revenue, therefore no costs of goods. But there is R&D expense, which makes sense. Their clinical trials expensive, and there's SG&A expense. So they're losing, at least last quarter, they lost around $60 million. All right, any interest income? Yes, a little bit. Oh, they have interest expense actually. So 1874 plus 357. So that debt, of course, comes with some expense. A little bit of tax expense there too. All right, now we're set. Let's do the same thing for last quarter. R&D 25,276. SG&A 12,631. No interest income. A little bit of taxes there, and we're good to go. So I'm just going to eyeball the cash flow statement, and you can see cash flow is a little better than net income in both periods. See, sometimes you get more information than you usually get. Combination of donepezil and a peripheral muscarinic antagonist. And then what's 104 is rivastigmine plus a muscarinic antagonist. Rivastigmine plus muscarinic antagonist. Okay. And they're testing them for AD and LBD. So we didn't update the front page last time for a few of these, but we did that now. I'm just going to glance through this relatively quickly. They did a loan with Hercules. That's where the debt comes from. Family members. Second product candidate is an inverse agonist for 5-HT2A inverse agonist. So, I don't know if you noticed that while scrolling, but I certainly did. And again, they describe the phase 2 versus phase 3 a little bit differently here, which is kind of interesting. .5% royalty. Arena Pharmaceuticals. That's where they got that neotanserin from. So write that down. risk factors. All right, so we kind of got through this 10Q filing again a little bit quickly, but I think that's fine. Most of our analysis should be actually on the medical clinical side. So let's go ahead and open up PubMed. And what we're going to do is, since we know that this used to be called GSK 72457, I actually think we should do a search for that. And maybe that'll get some more results. And we got 11 results here. So let's see if we just search for that number sequence. I wonder if there's more or less results. It's kind of asking us to look for something else. So maybe if I do this, that'll work. Okay. So we've got 16 results. And what I like to do in a situation like this is, it's a little silly, but let's get all of these in text, and I'm actually going to make a tab called literature. I'm just going to paste them real quick. And so as we paste them, we can read them, and we can get each paper and start reading them. But it's fun to kind of look from the first paper and go backwards. So 5-HT 6 receptor antagonist as novel cognitive enhancing agents for AD. This is a 2008 paper. The other thing we should do is look at GSK's patents. So I'll do that in a second. Here's Sertindol. Sertindol. I wonder what that is. Sertindol. Just looks like a classic atypical with the D2 antagonism. Notice the papers mention it. Looks like it's pretty potent at 5-HT6. In fact, it's more potent at 5-HT6 than most of the other receptors. Although it's got D2 and it's got 2A, which are the classic kind of atypical antipsychotic stuff. Alright, so they have more Sertindol papers. A little strange. And now finally you've got this Maher-Edwards study. So this is going to be really important. Let's read the Maher-Edwards study, number 13. Any clinical trial publication is going to be crucial. And this is the 357 patient study. So this is a brand new phase 2 study basically, which we did not look at before. And let's take a look at that. Need that PDF though. Where's that PDF though? Purchase PDF. I am so cheap, and I believe information should be free. So I am going to quickly see if this is on my favorite information freedom website. This randomized, double-blind, placebo-controlled study investigated the efficacy and tolerability of 5-HT6 receptor antagonist SB 742457. Let's see. Publish, incur, pulse, research, Maher-Edwards et al. 2010. Alright. Come on, Sam. So there is a statistically significant pretty close in all these studies. It's kind of interesting, right? You see there's some efficacy here. Still can't get the PDF. So what we'll do is we'll buy it. That's probably the easiest thing to do. But there's some notes here nevertheless. So the mean treatment difference from placebo to week 24. Let's look at they did 5, 15 mg, and 35 mg. That's pretty important there. As a 24-week study, co-primary endpoint CIBC and ADAS-Cog. P-value was 0.135. And what's kind of interesting is if you look at this study, you got roughly a 1.6, and this study you got a 1.3, and here you got this really bizarre kind of result of 0.7, positive 0.7. So you got two studies in a row that kind of give you a similar result, and then one study that's kind of out of left field. And that's somewhat interesting. And on the CIBC, it was a 0.6 benefit. So that's somewhat interesting. So let's keep going here. This is a so-called review paper. Review papers are a little less interesting than clinical trial papers, right? Well, here's another Maher-Edwards paper, published a few years later. Let's take a look at that. Maher-Edwards, a randomized placebo-controlled study. All right. 198 patients. But you could just write the paper, I don't know if they're published in. Geriatric, psychiatry, Maher-Edwards, all 2011. And you know, we should call Dr. Maher-Edwards as well. See what he or she thinks. Let's look at the design of the study. Okay. They're 35 mg placebo versus 35 mg versus Aricept. Pretty interesting and valuable study schema. Let's see, CIBC and ADAS-Cog. All right, so the ADAS-Cog was negative 0.4 and negative 1.2. So you can see that donepezil did quite a bit better than the drug, than interberdine, but both were better than placebo. They don't give p-values here. But you have a confidence interval. And the confidence interval includes the the upper bound of treatment, negative treatment effect. Okay, and they have an NCT number here. 003-48192. Okay. Let's keep going. CNS effects, the interaction of risperidone and 457. This is called a DDI drug-drug interaction trial, which is not too interesting. This looks like another review article. And this is a preclinical rat study. This is an in vivo study of LUAE60157, which, from my experience in pharmaceuticals, that is a Lundbeck drug, and we knew that Lundbeck was competing with this product. So this is a 5-HT6 antagonist as well. This, I think, was their arch rival, if I'm not mistaken. Let's see if we can just pop this in here. Ah, yes, iloperidine. Idalopyridine. Idalopyridine. We should make a separate page for idalopyridine, because if one drug works one way, if two drugs have the same mechanism of action, they should theoretically, not always, but theoretically they should have the same results. And if they have different results, there should be some scientific reason why we can explain those different results. Idalopyridine, not pyridine. Idalopyridine. Idalopyridine. Here we go. Now let's put it in the intepridine. They have the same suffix, that's how drug naming works. Idalopyridine. Idalapyridine is another way to say it. Idalapyridine. Okay, so let's type in idalapyridine, and it used to be known, let's drop that down, as LUAE 58054. 58054. Search just for idalopyridine, idalopyridine. This is what we get, a bunch of animal studies, and we should make the similar list. Excuse me. But here's the latter study. This was published in Lancet Neurology. Phase 2B. Latter. N equals 278. Alzheimer's patients, week 24, ADAS-Cog went up in placebo and down in iloperidine for a 2-point difference, p-value less than 0.01. Very nice. So let's see, what's the difference there? 2.16. 6. Placebo subtracted benefit and ADAS-Cog, p-value 0.004. Very, very, very robust result. And interestingly, they did better in donepezil treated, so this is maybe like a background of, they're all on donepezil, maybe. And let's see if we look at intepridine, these patients where the study wasn't working, they were not on donepezil, and maybe they were in these studies, so that could be the reason why it doesn't seem to work here, but it does seem to work here, and even here. So you got 4 studies, and 3 of them seem to work, and 1 of them didn't seem to work, although I'm using the word work kind of liberally. And for alloparidine, this was NCT 0101, 9421. And we should get structures here as well, and maybe see if we can get a 5-HT6 model. It's a little hard to model GPCRs. Therapeutic potential against obesity. And so the study's failed, as we know. Let's see if the Lundbeck folks put out those results or not. Maybe we search for 58054 instead. No study results posted. This was the Starbright study. Phase 3 flop. Starshine. Iloperidine showed a weak efficacy profile as neither of the 2 doses used in the study met the primary endpoint when added to donepezil. In addition, the secondary did not separate either. Safe and well tolerated. Find not just the press release, but maybe even the presentation or something. Doesn't look like it. All 3 phase 3s failed. Although I'm not sure. Let's see. so the reason this is important is, no two drugs are alike, but if these guys had a positive phase 2b, but then they had 3 phase 3s that all failed, it's hard to imagine that interbridine would have a different, more successful fate. These are more animal studies and in vitro studies. And this is the 5-HT6 in vivo study. I actually like studies like this. So let's look at study 7 for interbridine. Iloperidine, I should say. Oh, no, interbridine. There we go. Study 7. So this is a receptor binding study, in essence. And basically what you do in a study like this is you try to figure out how potent your medicine is. Let's take a look. Connection refused my butt. So sulfonamide, let's copy this. This is iloperidine structure. Learn a little bit about the receptor. Exclusively expressed in the CNS. Let's look at the abstract. So let's also get this down. Okay, got that all down. So So this is 8,4-methylpiperazine. That's this guy. This is a piperazine. 1,3-phenylsulfonyl-quinoline. This is the quinoline. This is the phenyl. This is the sulfonyl. So this is pretty simple to pump in and piperazine. And so we got a 0.2 nanomolar affinity, which is pretty interesting. That's very, very potent. Usually can't get less than 1 nanomolar, so really, really impressive. And good selectivity, but not, it doesn't not have an effect on 5-HT2A. So pretty potent at that receptor as well. .2 nanomolar Ki. This is 60157. So this is a, this is 60157, not 58054. So hold on a second. So this is a so-called tracer compound. So you do use this to study the binding of other compounds. Did a lot of work with syrup before. It's a good organization that does this kind of work. So we were able to, here we go, determine the ED50 NKI of these drugs, so including 457. So we got here is that 054 is 0.6 nanomolar, and 457 is 0.15 nanomolar. So they're very, very, very similar. You can see that 457 or intepridine is about 3 times more potent than idalapridine, but that doesn't, in the world of pharmacology, that's basically the same. You're really looking for orders of magnitudes of difference. Okay. So that's sort of the purpose of this study, is to show the affinity. And you can see that the atypicals don't have too much affinity for 5-HT6 other than a handful of them, including sertinidol, which folks have mentioned. You have the radiography, which shows the distribution in different parts of the brain. So this is, as I said, a tracer molecule. And here you have these, what do they call these? Schneider, I forget what they call these, Schild curves. Schneider. All right, so this is useful. It's very useful to know what, needless to say, what the affinity is. So the inhibitory concentration is less than a nanomolar. This is a very, very, very potent medicine. One of the things you can also calculate, let's say for aliperidine, is the log P. So you always need a log P. Let's go to PubChem, which is its own thing, and let's see if they have intepridine here. Yeah, there she is. Yeah, this is it. Intepridine. It's 353 molecular weight. Which is small enough to get into the blood-brain barrier. Let's get that structure. It's also, this is fascinating, huh? Wait a second. This looks familiar. What the heck? Come on. Well, it's not totally uncommon for... What the heck? No way. No way. Holy moly. There's no way this is right. This is a bigger finding than the Clinton emails. We all know Hillary Clinton's a crook, so that's not really much of a finding, is it? I am stunned. I am flummoxed. Right, log P is 2.5. And let's do the rest of Lipinski here. 1 hydrogen bond donor, 5 hydrogen bond acceptors. 3 rotatable bonds. Okay. This plot has thickened, friends. This plot has thickened. Alright, I think I copied and pasted the wrong molecule, that's why. So, if you look at, I was about to have a freaking heart attack. Look at this paper. Oh, thank God. That was about to be the craziest finding ever. So if you look at this paper, this is the tracer. This is not. Whew. That was going to be wild. This is the tracer. This is not 58054. This is LUAE 60157. And obviously, for any of you playing along at home, if this drug failed, then this drug is going to fail too. So they basically made a tracer out of, in essence, out of 457, out of intepridine. They designed this tracer molecule, which is the same, with the exception of one methyl group, the same as 457. And they didn't do the phase 3s on this, they did the phase 3s on 58054. Otherwise, we would not have a question as to whether or not intepridine would work. So that was a very scary, very, very scary. So let's move this guy maybe here. Let's put this guy here. Alright, so this is obviously very lipophilic as well, but let's get the statistics. And it's a very, very, very different molecule, needless to say. Let's get the... Okay, so this is a little bigger at 398 molecular weight. 2 hydrogen bond donors. 7 hydrogen bond acceptors. 9 rotatable bonds. Much more than the 3 that we see with intepridine. We've got a log P of 5, which is enormous relative to intepridine. What is that, 100 times the brain trafficking, basically? Or at least the partition coefficient. Some people like to look at topological surface area. I don't know if that matters too much. Let's take a look here. This is not the right molecule. There we go. 2,6 fluoro. This is the 2. No, this is the 6 fluoro. This is the 1 indol. Here's the ethyl. Here's the amine. Here's the benzyl. Here's the tetrafluoropropoxy benzyl amine. But hopefully that you can see that. 2233. This is the 1. So 2233. Very flexible molecule compared to intepridine, which is pretty rigid. Okay, so that was I was about to run out and start shorting the stock, even though it's midnight. But that's why you got to double check stuff. This is not the Lundbeck compound. This is the Lundbeck compound. Very, very, very important. All right, so we have a little bit more information. Again, this is still very concerning, and the fact that the affinities are almost exactly the same is also very, very concerning. So let's go back to the literature. Again, we have this rat study. You say there's some static? Yeah, I thought we hit it big. All right. Now here's this AVN 211. What the heck is that? Aveniro Pharmaceuticals and our farm. All right. There's another 5-HT6 group here. Let's see who these guys are. These are IMM Inserm, or Montpellier, I guess. So they made a different moiety. They got it to about 3 nanomolar. How are these guys? It's the medicinal chemistry department in Poland. Alright, so not a whole heck of a lot here. We got 2 extra studies to evaluate, which is very helpful. Both papers not easily available, but we can get them. So again, I want you to focus on this ADAS-COG here, where it's worse, I'm sorry, here, where it's worse than placebo. And then here, where it's better than placebo. And then again here and here, where it's better than placebo. These are the 4 data points we've got. Only one is statistically significant. This is, in fact, worse than placebo. This is better, this is better, this is better. These two, if you just looked at these two, you'd think that the study would work. 357 patient study is a little underpowered, so if you made it a little larger, maybe that p-value would drift a little bit in the right direction. And here, again, this is sort of a very weak signal. I mean, it's less than Aricept was generally considered to be a very weak drug itself. The thing about how p-values really work is that you're either sort of have a signal or you don't. And I just sort of, let's sort of do some statistical modeling here for mindset. So if you have 1,315 patients, you'll have 650 or so per group. And now let's try to get some of this statistical analysis down here. So let's go back to this study. You have some variability that we can use here.
So these are the old studies. I thought there was a published study that we pulled up yesterday that actually had some of these results. That's why you try to write these things down, isn't it? Let's see here. Maybe we type RVT 101, maybe that'll work. Get the same result. Well, let's pull it up. Have this. I think we put it in here, right? SB 742457. Okay, well it might be because that SB 4742457. So we should also query that in here. SB 742457. Usually PubMed's pretty good at this indexing, but not always. So you can see this. Maybe I just missed it in the list somewhere. No, I don't see it. This is a 2015 paper, so it should be right here, right? I don't see it. Crazy. Maybe with no hyphen. Still don't see it. Crazy. Maher Edwards published it too. This is a GSK employee, Maher Edwards. How wild. Where'd I get this mystery paper? So now we have a standard error. We can do a little bit of p-value analysis. So 24 weeks. 35 mgs. Mean was 0.4 and the standard error was 0.62. And placebo, we got a similar standard error. So maybe the standard error is going to be like 0.6. And so from here, we can actually do a little p-value calculator. And GraphPad does it the best. So GraphPad, you can calculate p-value from, let's see, what should we do? We do it like this. I think we can do it like this, or do we do it, let's go back further. Categorical data? Nope, it's continuous data. Let's do a t-test, which is really, really simple. All right, so what we'll do is enter mean, standard, error. I think this will work. All right, so let's make this in Teperdine, and let's make this placebo, and let's see the mean is, let's say it's 6, and for placebo it's say 7. Standard error 0.6.6, 650 for 650. Calculate now, and this would fail. I want to make sure that this SEM and the SE are the same thing. I think they are. I think they are. Yeah, same thing. So if you, let's see, bump the treatment effect to say, and again, the 8S cog, higher is worse. So let's say you got 2 points. Let's see, I don't know what the 8S cog 11, or is this 8S cog? Let's take a look. 8S cog 11. All right. So let's see, let's try 2.8S cog with standard error 0.6. Well, let's calculate that. Then we have a 0.0186. 3.8S cog with standard error 0.6. P-value is 0.0004. Okay. Let's do a few more things. So what's interesting about this is that even in its best day, intepridine had a treatment effect of 1.6, which would be sort of between statistically significant and not statistically significant. Let's make sure we're not being too mean with respect to the standard error. And we see here, let's see, 8S cog over here, let's say, at week 24. Well, the standard error is a bit better. At 0.4, so let's recalculate them with 0.4. And in fact, as you can see here, we should be able to get their p-value or something close to it. Obviously, you have to do all kinds of other Cox, various different adjustments to the ANCOVA, etc., and ANOVA. If you're statistical mavens, you'll know what I'm talking about. But let's do a 1.512 because that was the actual treatment effect seen here. Now let's do it with a 0.4. If you average these two, it's 0.43. So let's try 0.43. So let's start with, let's see, so mean 6 and 7. Standard error is 0.043, 0.043. And let's do 650 apiece. Calculate now. Just want to make sure I have all this right. Looks good. Okay, p-value is 0.103. If we did 5, p-value is really good. It's 0.0001, and needless to say, at 2 or more, it's ooh, that's for 2. So let's do 1.5. 0.0138. Thought that was a little too good to be true. All right, and now, of course, for 3, there's no doubt at that point. So you can see how much standard error matters, of course. And what's interesting here is that if you pick the standard error of their study and you calculate the difference, you get very close. We got 0.0138 and they got 0.012. And in fact, if we really want to test our graph pad skills, maybe we'll try to do this exactly. I don't know. 193 for placebo. This is 0.14. This is 0.1. 4 versus 0.1. 5. And there's 1.5 difference. I don't think this is going to give us the right result. Oh, 0.014. So it's very close to their p-value. And there's probably some ANCOVA changes that would fix that. So anyway, it really depends on whether it's going to be, the standard error is going to be sort of closer to this or closer to that. And then we'll have to sort of think about let's do one more of these. I remember patients drop out as well, so that you lose a little bit of power. So you can see here, even if the variance is high, you're not going to hit statistical significance. So that standard error is often underappreciated. And you always lose a little bit off of the desired enrollment. Patients drop out of studies and you have to do LOCF or BOCF. Last observation carry forward, best observation carry forward. It's very important to statistically model a study to see if you're going to hit your endpoint or not. And these guys are pretty smart, and that's why they added many patients, 1,300 patients, to boost their chances of success. The problem is the effect size, right? I mean, the best effect size they've ever had was 1.6 and 1.3, and we're totally ignoring the 0.4 and the bad 0.7. That's in the wrong direction. If you average all of those, which isn't a good way to do it, but let's do it for fun. The average is negative 0.645, which is really, I mean, if you think about an 11-point scale, 0.6 is a 5% improvement, which if you have Alzheimer's is really not much of an improvement. Now if you take the best one, it's over 15% improvement. Yeah, I have a theory on the Argentinians as well. So yeah, if you assume that they'd have to sort of they'd have to repeat their best performance ever, which they got in this this study, and then they'd have to come in at the lower standard error. So one of the things that we could do is look at donepezil. Let's actually go into the here. And what we'll do is we'll go back in time. There's a lot of a ton of papers, but we'll have to go back in time and see if we can get the efficacy study. So here's one of them, neurology, 1998. I wonder if we can get this paper. Let's see. Might be able to. Here it is. It's probably one of the first trials of donepezil. There's the ADAS cog. You can see the standard error. I'm sure we'll get the actual number, but you can see placebo drops 1.7 or 2, and drug goes up about a point. So you get about 2.5 or so points here. the ADAS-Cog, but it's not the ADAS-Cog 11. Keep that in mind. I think that might be different, although I wonder if the ADAS-Cog 11 is actually around 30 points. Let's look one more time at that. And let's also look at the paper that we just saw, because I think that'll be really interesting. So I think there's baseline here. Yeah, here's the baseline. So this is going to be really important. Huh. Okay, let's take a even closer look at this data. 35 mg baseline. Let's wrap it around here with placebo baseline. That's right, your placebo 24 week change and 24 week change. Alright, so the placebo's baseline data was 27.1. And 35 mgs was 27.1. So this is a really well done study, right? Remarkable. Let's try it for study 1. Sometimes you get sicker patients at baseline, and you really have to make adjustments. So let's see here. Placebo here was 29.1, and Tecridine 35 mg was 28.2. That's only a point difference. It's pretty close. You can see donepezil baseline. They were really much better off, it looks like. Baseline's the same, of course, no matter what. Looks like at 26.2, I think I got that right. Were they worse off? Yeah, worse off. You're worse off with the lower score, right? Clinical decline. No. Worse off with higher score. Mean change from baseline. Yeah, that's right. Higher score is worse. So, conceivably, this should have biased the study. You could look at it two ways. A sicker Alzheimer's patient can't get any worse. I don't know if that's true. All these studies do progress, because this is mild to moderate, so keep that in mind. So I think you can reject that. I think basically if you have a lower score baseline, you've got healthier patients that should do better. Although rate of decline does make a difference. I don't know, it's hard to interpret. But here you have almost identical baselines, so if not identical. This is the standard deviation of the patients at baseline, which I don't think is as relevant as the standard error. So let's look here. All of these patients had a score of about 27-ish. And here's the mean change with the standard error. This is really valuable. Let's see if that's the standard error. Mean change, plus minus, standard error measurement. Great. So you can see here that you may not get that better standard error. That standard error was 0.57, 0.56, and 0.64. In this paper, Rogers et al. Neurology, 1998. Friedhoff was actually works at Axon now. And you can see again at 24 weeks a very powerful change. 2.49 and 2.88. And the p-values were wildly positive. Again, Interbreeding has never had any data close to this. The best they had is that 1.6 that I showed you, and this 1.3, which is about half. Around half, but you're also ignoring the 0.4 and the negative, the really negative outcome of 0.7. But again, even if you sum all of them, if you average, I guess, throwing out the worst one, sometimes it's fair to throw out the worst and throw out the best and average the rest. And that's about a point, which is still about half the size of Aricept. Did I look at the right standard error? I think I might have looked at the wrong one. Here's the right standard error. Where was I looking at? Let's look at that one more time. 5.6, 5.7, 6.4. Where'd I get that? 5.7. Oh, that's at 30 weeks. The 30 weeks is going to be more variable than 24 weeks. So let's go 49, 51, 51. Well, that's a little bit between, isn't it? $20, 49, 51, 51. All right. All right, let's try GraphPad one more time. I know you might be getting sick of this, but let's do it with those values. All right, so maybe it makes sense to do the means that are more realistic. 27, 26 versus 27. Standard error 0.5, 0.5, 650, 650. This is a failure. It's about the in between the p-values that we expected. We make it a little better with 25.5. And now p-value is 0.03. Now we give it 25. P-value is 0.0047. And here, really good result. P-value is less than 0.001. So obviously, if they can get 2 points, it looks like they're Gucci, technical term, on all of them. And we don't know what study will show up. Will it be the best case scenario here, the Aricept scenario here, or the worst case scenario here? But we do know that they never got better than 1.5 or so. So I think it'd be a tough bet to make, if you ask me, that of the 4 results, they tried this experiment 4 times, you're basically betting that this 4th, the best of the 4 will repeat itself. Because if anything else happens, no bueno, as you can see here. unless the study is really, really tightly done in the sense that, say, 1.3, and the standard error is close to 0.4, you get a p-value of 0.02. So you can barely meet. You'll barely meet it, but you lose a little power because I'm using 650, which is 1,300 patients. But you usually lose a little bit of power due to a couple of different reasons. I mean, one is that they have dual primary endpoints, which is problematic because usually you have to split your alpha between the two, in which case our p-value would have to be below 0.025, which, this wouldn't be enough. So looking at the error sub results is helpful. It really frames everything here. And the other thing that's really, really important is that when ADAS-Cog, when the baseline, let's look at the baseline, you've got a baseline of 26 or 28. And you're adding 1.6 to that. That's in the best case scenario. You're talking about a 6% response. If we take the second best response, which is 1.3, you're talking about a 5% response. And then the third best response they've got, which was 0.4, you're talking about a 1% benefit. So this is the benefits we're looking at for the patient, which, I don't know, if you told me my Alzheimer's would slow down by 5 or 6%, it's always, nice to get a slower disease, but in most illnesses, cancer and so forth, you're really not getting a whole lot by saying you have a 5% improvement in illness. And of course here we have the negative response with 0.7. And so the other way to look at it is something called Cohen's d-value. And in essence, Cohen's d-value is clinical effect. So it's basically the change over the standard deviation. And here, you have a change about 1.5 points. You have to ask yourself, what's the standard deviation? Well, we know what the standard error is for this 0.5 and 0.43, etc. But we can work backwards to get the standard deviation. I would venture that the Cohen's d-value here is very, very, very low, because donepezil is Aricept. It was a $3 billion medicine, which is why there's some hope here. But the clinical relevance is really really sort of limited even if they get a 1.5, because most people question the clinical relevance of Aricept. And you can see that gets you about 2.5 to 3 points. So 1.5 to 2 points here would be, again, Aricept sold $3 billion, so it would be used. And there could be some synergy to give both medicines, and maybe you get a 10 or 20% benefit. But it's everyone agrees this is not a miracle drug. Nobody thinks that. It's just a question of will it get approved. And getting approved is the name of the game. Most doctors will prescribe a new Alzheimer's drug. Again, a couple things we learned here is that the pharmacology, that you have a subnanomolar medicine here and a subnanomolar medicine here, and with the Lundbeck medicine, and it failed 3 phase 3s. With one good phase 2 with about a 2-point benefit in that phase 2. So the fact that 3 larger studies in that phase 2 failed to replicate the result in phase 2 is really telling. And it does tell you that you can't cherry pick the best of the 4 here. You really do have to look at all 4. And if you look at all 4 with a sober eye, you'd say, well, one worked. One was so bad that it really, I think, kind of negates the idea that this one worked. And then you had two more that failed. This confidence interval overlaps. This p-value is 0.135. So I still have more work to do, but we've looked at pretty much all of the clinical data, I think. We have more to do, obviously. One of the things we want to do is hear from the company. We looked at the major drug that's similar and has had a bad experience. We looked at a lot of the published literature. And yeah, I'd say that things are looking not so great. For Axovant, maybe nilotanserin is what saves the company. What happened with Medivation, which was Dr. Hung's last company, is he had one drug for Alzheimer's that nobody believed in, or most people didn't believe in, called Dimebon. And they had another drug for prostate cancer, and it's a prostate cancer drug that ended up being worth billions and billions of dollars. So maybe it's nilotanserin that's worth billions and millions of dollars. So I think that the market's not going to count Dr. Hung out very quickly, because he's a pretty great entrepreneur, and they did really well with, all you need is 1 out of 2 shots, right? So this stock could drop to say $10, but it doesn't mean that the company's all over. That's why I think it may not drop to $5. So if you're buying the $10 puts, right, the $10 puts, $10 puts is the right to sell at 10. Well, what's the right to sell at 10 if the prevailing price is 20? Well, it's not worth anything. The put isn't worth anything. Because if the prevailing price is 20, the right to sell it at 10 is not valuable. The right to sell it at 10 is only valuable if it's trading for less than 10, right? Now these puts right now, $10 puts are trading at $5. So basically they're only valuable, they're not trading at $10. Let's see, what are they trading at? I think they might be trading at $10. At $5. Doesn't make too much sense. There might be $15 puts. Let's take a look. Let's go to the videotape, as an old sportscaster here in New York used to say. Type in Axon, go to options, go to October options, because that's when the data will be out. And let's go to puts. The $10 puts are, where are they? $2? There aren't that many puts outstanding. That'll change. That'll change. Isn't that weird? Maybe Yahoo Finance just doesn't know. Let's take a look at the 10 puts. I think I was looking mostly at the 15 puts, so those are $5. 15 puts for $5. Okay, so they've been around $5. They're about $5. Okay, so the right to sell at 15 is not worth anything if the stock's trading at 20. It's not worth anything if the stock's trading at 15, right? Because you can just sell in the open market. You don't need a special contract for that. But if the stock trades at 10, the 15 puts are worth $5, if that makes sense to you guys. Because you have the right, even though the stock's trading at 10, you have the right to sell it at 15, and so that right is worth about $5. No way Carl Icahn passed away. No way. That'd be so sad. That'd be so sad. He was in pretty good health last time I saw him, so doesn't look like he passed away. So maybe we can ban this TMV guy. That'd be so sad. He's a hero of mine. He's very old, but I didn't think he was close to death. All right. Anyway, so the point is that if you buy these 15 puts, you only make money if the stock goes below $10. And I wonder if the stock will really go down, because even if Intepridine fails, there's still nilotanserin, and you still have David Hung. And you still have 2 more medicines here. So I think the stock might drop It will definitely drop. But it has to go below 10 if you buy the 15 puts. Now, if you short the stock, you'll make money if it goes to 10, but you're also taking unlimited upside risk, whereas on the puts, the most you could lose is the put contract itself. So you just have to sort of think that through. Of course, there's 20 puts and 10 puts and so forth, but what you really have to get at here is how much will a stock go down? Say it does fail. You know, this is reductio ad absurdum. Assume the conclusion instead of the opposite, which is basic deductive reasoning, which is assume the givens. And so if you assume the conclusion that the study fails, where will the stock go? And you could argue that it should go to its cash value, right? And its cash value is somewhat limited. It's only $2 of cash, and I don't know how much nilotenserin is worth. Say that's worth $500 million. Then I can add 500 into this, and maybe the stock will go to 7. And you can see that people may think, okay, well, nilotenserin's worth $500 million, or David Hung's worth plus nilotensrin are worth $500 million, but typically these stocks don't go as far down as you might think. So the 10 puts or the 15 puts in this scenario, say the 15 puts, which cost you $5, they'll only go to $8. So you'd only make 62%. Well, that's a lot, obviously, but there's no guarantee that it goes to 7. It might go to say 10, right? In which case you don't make any money. And nobody has the crystal ball to say, oh, it's definitely going to go to 7 or it's definitely going to go to 10. That's a really tough thing to say, and the markets are so wild on a day like that where the results come out that you don't know exactly where it would go. So I think that the study will probably fail, is my guess. Based on the work I've done for this video series, I'm not sure I have enough information to short just yet, but it certainly looks like it's going to fall, or fail, I should say. But I don't know how you make money off of that, because if you short the stock and you're wrong, you lose an absolute fortune when the stock goes up. So that's sort of an important consideration, and there doesn't appear to be a put option contract that's really valuable. So if you buy the 20 puts, let's go look at that. If you buy the 20 puts, they're $8.50. So they're already pricing the stock going to $11.50, right? So whatever difference, every dollar below $11.50 it goes up, you make $1. So that's your break even, is $11.50. If the study fails, it has to go less than $11.50 to make any money, which is a pretty tough thing to do. There's a few other strategies you can take, a bull spread, a bear spread, but they all have the same return, in essence. So, I don't know, really tough place to be. You can sell volatility, which seems to be, I think, a good idea. So if you take the 20 at the money, it's $8, right? And the 20 here is $8. So if you sell both contracts, as long as the stock moves less than $17, you make money. You short both contracts, collect $17 doing so. You make money as long as the stock stays in the bound between 3, greater than 3, and I guess less than 37, which I would say is very, very likely. So maybe you sell, you sort of sell both, contracts. That's often when there's a lot of vol, that's often what's best to do anyway. But the there's not, the open interest, I cannot be accurate here. I'm sure the open interest is much, much higher. That's why I use Bloomberg for that. But it's a fascinating situation, obviously. It's captivating a lot of people. If you're feeling crazy, the 7.5 puts, if the stock really does go to cash value, the 7.5 puts would be would allow you to make quite a lot. You make 6, 7, 8 times your money. I just don't think the stock's going to go to 2.5. I don't even think the stock will go to 5. So the 7.5 puts would be worthless. The 10 puts would be worthless. I think there's some. The last time this happened, people did really well when David Hung's. When you bought the failure after Integrity failed. So I'm sort of skeptical that the stock's going to fall apart. So maybe it's best to just short the stock and sell the call spread. So it's a very bearish bet. Not sell the call spread. Sell the straddle, which is what this is known as doing when you sell vol at the same strike. So anyway, I hope this was helpful. We got a lot more to work on, but this is part 2 of the Axon, how to look at a clinical trial situation. Thank you so much.